Welcome to our fall Dean's Distinguished Lecture, and our special guest is Dr. Aimee Edinger. Just as a little bit of background, I know a lot of you have been coming to this since its inception. This was really started as a great way for us to, in the school, learn about each other's research and to also share that with the members of our community. And I'm really happy that Dr. Edinger agreed to speak for this fall lecture. She is just a remarkable scholar and scientist who has won just about every accolade that you could win. She has won awards for her teaching, both from the school and from the campus. She has a distinguished title as a Chancellor's Fellow, which is a designation that's meant to identify scholars who are distinguished and on a track to continue that distinction and earn even more recognition in the future. She is an inventor and has started her own company, and I think is listed as an inventor on 7 patents or so. An outstanding scholar, and I've gotten the chance to get to know her quite well because she did double duty as equity advisor for the school, serving 6 years on behalf of the School of Biological Sciences, and she was just truly passionate and a genuine delight to work with. I have to say I've always admired her values and the strength of her convictions, and I miss her in the dean's office, so hopefully we'll get her back there soon. So she's going to talk about, let's maybe move this slide to her presentation, Redirecting Cellular Traffic to Treat Cancer and Obesity, which I think you all will agree is a very important topic in our society today. So let's welcome Dr. Aimee Edinger.
So thank you all for coming. I'm super excited to have this opportunity to share our research with the BioSci community. I'm very happy to see some of my students here and colleagues who have very busy schedules, community members. I'm really happy to see you all here. Thank you so much for letting me share my story with you. I have a quick disclosure. As Frank said, I started a company. Hopefully you'll understand the name by the end of this. And I also do some consulting. Okay, so based on the title, I think you understand that the goal of my lab is to learn how to control the flow of traffic within cells. And this is an important and meaningful goal because the cell is like a small city. Different activities are compartmentalized in different organelles in the cell. So you have the nucleus, functions kind of like City Hall, issuing orders, controlling what's going on in the cell. But it's also kind of like an archive. The genome and the epigenome have volumes of information that the cell uses to make the right decision. Mitochondria, well known as power plants, provide the energy for cellular processes. Lysosomes are the waste management company, handle recycling and disposal. ER and Golgi are like a factory that makes the protein and lipid products that the cell uses to do its business. And the plasma membrane is kind of like a city wall. It allows the cell to be different from its environment by carefully controlling what can go in and out. Now, for this city to function, materials and messages need to travel between all these different compartments with high fidelity. And so to do that, the cell uses small membrane-bound vesicles that kind of act like Amazon delivery trucks. Now, we can all reflect on our own lives to understand why delivery systems are so important. So FedEx is why all of us who got the COVID vaccine were able to get the COVID vaccine. It enables my collaborations with researchers in Canada and Australia. Out of stock, common during the COVID era, the UPS strike last year, all the packages that are accumulating on our porches as I speak. These are all tangible reminders of how important deliveries are for us to go through our daily lives. So we're interested in sphingolipids because they direct cellular traffic and thereby control the delivery of different things around the cell. We're particularly interested in 2 natural sphingolipids. Phytosphingosine is found in yeast, and sphingosine is the mammalian form. Now, these sphingolipids reroute vesicles to new locations in the cell to help them deal with stress. For example, if a yeast is in conditions that become too hot, it makes phytosphingosine as part of an adaptive response. These yeast here are expressing a fluorescent tryptophan transporter on their cell surface, so that's why there's a ring around them. Amino acids are highly polar molecules that can't just diffuse into the cell. They can only cross the city wall through transporter gates. Phytosphingosine downregulates those gates and restricts access to nutrients. This induces a hibernation-like state that helps the yeast survive stress. This pathway is preserved in mammalian cells. These are prostate cancer cells, and again stained for an amino acid transporter, downregulated by phytosphingosine and sphingosine. They get trapped in these intracellular puncta. Now, the way sphingolipids do this is by setting up two different roadblocks. So this cartoon shows you nutrient transporters in the plasma membrane. They're constantly cycling into the cell and out back to the surface. They're rapidly returned to the plasma membrane unless they need to be repaired. The first roadblock is here. So by blocking that recycling step, you end up with transporters accumulating inside of the cell. Now, because this is a response to stress, the transporters are coming down to starve yeast into quiescence. You want to reuse them again when conditions improve. So we don't really want them to go to the dump. We don't want them to go to the lysosome. So sphingolipids set up a second roadblock here. So where these puncta are accumulating is actually this pre-lysosomal compartment. Now, given that these sphingolipids can restrict access to nutrients and starve cells, they might be a good weapon against cancer. Many of you are probably familiar with this quote. It's often attributed to Napoleon Bonaparte. An army marches on its stomach. And so what we wanted to do was use sphingolipids as a way to stage an attack on the supply wagons, to starve cancer cells to death. Now, if we want to do this, unfortunately, we cannot use natural sphingolipids. So by 6 hours, phytosphingosine is degraded and the transporters are back on the surface. Cancer cells express really high levels of the enzymes that turn over sphingolipids, so that's going to be a big problem. So I collaborated with a medicinal chemist, Stephen Hanessian, and we used a standard medicinal chemistry trick, which is conformational constraint. So we wrapped this part of the molecule into rings, and that stabilized it. Now, this compound, SHBC893, is named after Steve Hanessian. It's the 893rd compound that Bin Chen made during his thesis, so I'm going to call it 893. And you can see that when you add it to these yeast cells, it does the same thing as the natural compound phytosphingosine, but its effects last longer. If you put it on cancer cells, it also downregulates nutrients. So here we're looking at transporters for amino acids, sugars, lipids. They all end up inside of the cell, and the amount on the surface declines. So this means that 893 should be a very powerful starvation agent. It's relatively stable, and it disrupts multiple different nutrient import pathways. So we tested it in a cancer model. This is a genetically engineered mouse model that lacks two key tumor suppressors, P10 and P53, in the prostate. These mice get large tumors at about 5 months of age. If you treat them orally with our small molecule once a day from the age of 8 weeks, you can see the tumors are 82% smaller, and they're also less aggressive. So you don't really have to be a pathologist to see that the cells down here are more organized into something sort of like an epithelium, whereas up here in the controls, very disorganized. Now, importantly, 893 also works in tumor cells that are derived from patients with drug-resistant metastatic disease. So when these are grown as tumor organoids in 3D, you can see that the standard of care, which is called enzalutamide, really doesn't do anything. You see live cells with the calcine stain and no dead cells with ethidium bromide. In contrast, if you treat with a lower dose of 893, you see that you're able to kill these drug-resistant cells from patients. Now, at this point, most people are thinking, okay, I get it, kill a cancer cell, starve them to death. What about the normal cells? Aren't the normal cells going to starve too? And this is where I want you to think about bears in the winter. So normal cells, when they're under nutrient stress, go into a hibernation-like state where they're quiescent, catabolic, can survive the nutrient stress by keeping their energy use in balance. So they survive, but the cancer cells have oncogenic mutations that are constitutively driving growth. They've deleted or dismantled the tumor suppressors that allow hibernation. So these guys have a bioenergetic crisis under the conditions that normal cells can hibernate and survive. So this is our therapeutic index. And the data seem to bear out this model for cancer selectivity. So in those mice that were treated for 3 months with the small molecule, we did a blood chemistry panel to look at organ function. Nothing was really disturbed. While the cancer cells struggled to proliferate, the rapidly proliferating cells in the bone marrow, like the red blood cell precursors, white blood cell precursors, they were fine. They continued to produce hematopoietic cells. In the gut, where cells turn over every 5 days, things looked normal. So with these promising preclinical results, what we're doing right now is we're making some structural changes to the molecule to take it from something that works in mice to something that can be safely given to human patients in clinical trials. So a compound that starves cells, reduces nutrient access, that probably has multiple applications. While I was talking about cancer, you were probably thinking about obesity, since the dean introduced that idea. And obesity is a big problem in the United States. So when I was in college, about 20% of American adults were dealing with obesity. Today, about 50% of US adults are living with obesity, and there are even more dire predictions for the future. And I think a lot of people don't really realize how big the impact of obesity is. The cost of treating the complications, the medical costs of treating the complications of diabetes in the US are $260 billion a year. Because it causes things like cardiovascular disease, liver disease, type 2 diabetes, some cancers are associated with obesity. High BMI is actually second only to smoking as a cause of preventable death. And globally, it's estimated that obesity kills 5 million people each year. That is a huge number. That's on par with the number of people that died in the first year of the COVID-19 pandemic. So this is a very serious problem. Now, standard prescription, more exercise, improve your diet, it definitely has a place in the treatment of obesity. It will be important in the long run. However, we now realize obesity is a disease with its roots in biology, and we're going to have medical therapies if we're going to address this epidemic. So, what are the biological causes of obesity? One of them is malfunctioning mitochondria. When you eat a healthy diet, your mitochondria look like spaghetti. They're long and stringy, okay? Palmitate, which is found in things like bacon cheeseburgers, is a fatty acid that causes your mitochondria to fragment into meatballs. When mitochondria look like spaghetti, they're like a charged battery. They're able to provide ATP for cellular processes. But when they're meatballs, they're like dead batteries. No charge, as you can see with this dye that measures mitochondrial membrane potential. Spaghetti mitochondria operate normally, safely function. Meatball mitochondria spew these toxic things called reactive oxygen species that damage cells. So mitochondrial morphology responds to your diet, but it can also cause obesity. Scientists made mice that had spaghetti mitochondria all the time. And these mice, on a high-fat diet, stayed skinny. On the other hand, if you make a mouse that constantly has fragmented mitochondria, those mice have the opposite phenotype. They become obese on a standard diet. So this means that mitochondrial shape can actually drive obesity, can be a cause of obesity. But what does this have to do with sphingolipids? Well, it turns out that those vesicles that are delivering items around the cell actually deliver things that are critical for mitochondrial fission. And we don't really need to talk about the details, it's not important here. But these two roadblocks that starved cells, those same roadblocks should stop mitochondria from becoming malfunctioning meatballs. And that is what happened. So if you treat with our small molecule 893 along with palmitate, mitochondria stay spaghetti. They also maintain their energy charge, and they do not make toxic ROS. These are cells in culture. What about in mice? It also works there. So here we have a liver that's been freshly resected from a mouse and plopped on a microscope stage, and you're looking at mitochondria based on NADH fluorescence. This is a single hepatocyte here. This is the nucleus of that liver cell. And you can see the mitochondria kind of tubular. Maybe they're more fusilli than spaghetti. Appeal to Frank's expertise here. But after the mice have been eating a high-fat diet for 6 months, you can clearly see that there's a difference. They round up like meatballs or snowballs. And just 4 hours after you treat them orally with 893, tubularity is restored. You see something similar in the brain. So brain mitochondria are a little bit more wispy, but still kind of tubular. When the animals are on a high-fat diet, they round up. Treat with 893, tubularity is restored. In this case, I am showing you a little bit of quantification because the phenotype is more subtle. So on a high-fat diet, the mitochondria get rounder. When you treat with 893 on the high-fat diet, they go back to a normal level of roundness. And these neurons in the arcuate nucleus are the ones that control appetite, and we just picked a random area of the cortex. Okay, so mitochondria in fat are also made more healthy by sphingolipids. You may not realize that your fat plays a really important role in keeping you skinny. So healthy diet, nice healthy mitochondria in adipose tissue. The adipose makes a variety of hormones. One of them is leptin. Leptin travels to your brain in the blood and tells your brain, I'm full, and you stop eating. Now, in mice or humans that are on a high-fat diet and have obesity, the mitochondria become fragmented and dysfunctional, and that disrupts adipokine hormone secretion. What happens is leptin levels go through the roof. They go so high, the brain is like, stop, I'm not going to listen to you anymore, and it becomes resistant to these leptin signals. As a result, people are very hungry and they eat too many calories. So can 893 fix this? These are adipocytes looked at right after they were taken out of a mouse, and they look a little weird. So this big black circle in the middle is a giant lipid droplet in the middle of the adipocyte, and all the organelles are squeezed into this narrow ring of cytoplasm around the outside of the cell. But you can see the mitochondria are charged, they have a potential, and the ROS is in macrophages that are in between the cells. When you put these mice on a high-fat diet, their mitochondria in the fat become dead batteries and make toxins, but 893 fixes both of those problems. Does it fix the leptin problem? Yes. So in the mice on a high-fat diet, 893 restores normal leptin levels, and it also makes the brain stop listening again. So SOX3 is a negative regulator of leptin signaling, and in mice treated with 893, the levels are reduced back to normal. The leptin receptor is how the cells sense leptin, and you can see 893 increases levels of the receptor. So we have a compound that makes mitochondria tubular and healthy and restores normal leptin signaling. We decided to do an interventional study to see if you give this compound to a mouse that's obese, can you cause weight loss and restore metabolic homeostasis? So here you can see that these mice were given 893 Monday, Wednesday, and Friday, 3 times a week, and they rapidly lost weight even though they were eating a high-fat diet. These mice were not sick because we put them in cages with a running wheel, and they ran 2 kilometers a day voluntarily. And that caused even additional weight loss. So the mice are feeling pretty good losing weight. Because of social media, I have a feeling most people in the audience have heard of Wegovy or Ozempic, the GLP-1 receptor agonists that cause people to have a decreased appetite and lose weight. When you give that compound to mice, you get weight loss that's pretty similar to what we're seeing with 893. So it's on par with standard of care. This compound also restores metabolic homeostasis, so it reduces ectopic fat accumulation in the liver. This causes liver disease that can progress to cancer. It also restores normal glucose homeostasis. So here we give the mice an oral glucose load, and they can rapidly control the glucose levels, suggesting that insulin sensitivity is also restored. So these are good things that make me happy. At this point, we have a compound that hopefully I have convinced you does interesting things at mice that could have value as a treatment for lethal human diseases. What we need to do now is take it from a compound that works in mice to something that will be safe to give human patients. And to do that, this is called lead optimization. It's really helpful to know exactly how the compound works. So we tried to figure out what the protein targets of sphingolipids and 893 were. To do this, we collaborated with a medicinal chemist, Stephen Hanessian, and a proteomics expert, Pierre Thibault, to do this thing called chemoproteomics. And basically what you do is you take chemicals and you use them as bait to fish in a cell lysate to see what proteins bind. With these particular compounds, it's a little bit tricky because they're very sticky. This long hydrocarbon chain makes them bind to a bunch of trash, not just targets, but trash. So to separate the targets from the trash, we use a second kind of bait. These are structurally very similar, but inactive in cells. They don't change intracellular trafficking, and these should catch only trash. So if you look at individual proteins, ones that are caught by active compounds but not inactive compounds are probably your real targets. So we made a ratio for the 4,000 proteins that we found in cell lysates of how much they bound to the active compounds relative to a matched inactive compound. And the ones with the highest ratio are the ones that are most likely to be targets. This heat map shows every protein out of those 4,000 that bound at least 8-fold better to one of the active compounds. And you can see these 5 at the bottom are special because they bind better to all 4 of the active compounds in 2 different cell types, a hematopoietic cell type and a prostate cancer cell type. So these are our putative targets. PPP2R1A wasn't a huge surprise. That's the scaffolding subunit of PP2A. Everybody agrees that these sphingolipids activate protein phosphatase 2A. These guys at first made me sick to my stomach. So these are all importance. No one had ever linked importance to sphingolipids. This was kind of a shocker. There's no amino acid sequence similarity between these proteins, but structurally, they're strikingly similar. So all of the importance have a similar shape to important beta, and they overlap with the PP2A scaffold. So I think there is some kind of structurally conserved binding site here. So our first job was to figure out were our fish really fish, and to do that, we used this assay called the thermal shift assay. We made recombinant PPP2R1A and recombinant KPNB1, and then put them into this assay that measures conformational changes in protein. And this is based on this fact that heating proteins will make them unfold. So normally, hydrophobic amino acids are tucked inside of a protein. As you heat them up, they start to be exposed. This assay uses a dye called cypro-orange that emits light when it binds to hydrophobic amino acids. So as you increase the heat, you increase cyprobinding, you increase fluorescence, until the point where increasing heat causes the protein to aggregate, hiding the hydrophobic amino acids again, and then the fluorescence declines. So this point of half maximal fluorescence is what we call the TM, or the melting temperature for that protein. When a ligand binds to the active site of a protein, it tends to stabilize it. So it shifts the TM to a higher temperature. And the graphs I'm going to show you next, they're first derivative plots of these plots. And so the TM is now at the peak. It just makes it a little bit easier to see the shifts. Okay, so we took PPP2R1A, made in bacteria, pure protein, mixed it with our small molecule, and you can see that it caused a conformational change that decreased the TM. So that was kind of unexpected. It means that it unfolds the protein. But that was also kind of exciting because another group had shown that unfolding this scaffold actually activates the PP2A enzyme, which is exactly what these sphingolipids do. An important control is the inactive compounds do not change the conformation of these proteins, no thermal shift. We got very similar results for the importans as well. This is KPNB1. So because sphingosine and 893 slow growth in metabolism, we thought they're probably inhibiting nuclear import, inhibiting the function of the importans. Now, that kind of makes sense when you think about the nucleus is the seat of government, critical archive. You're going to carefully control what goes in and out of the building. So the nucleus is surrounded by a membrane that has these little dots in them. Those are nuclear pores. So those are protein channels that have this gel-like protein in the middle. So the only way that a protein can get through that gel and get into the nucleus if it's holding hands with an important. So importans are chaperones that bring proteins through the gate into the nucleus. We started by looking at the endogenous cellular KPNB1 in these prostate cancer cells, and you can see in the zoomed in image, it makes this nice ring around the nucleus. You can kind of see the little dots. Each of those is an individual nuclear pore. So KPNB1 is wiggling its way through that gel, making a nuclear ring. There's also some KPNB1 in the nucleus. When you treat these cells with 893, you lose the association with nuclear pores and you lose the protein from the nucleus. So this is consistent with 893 inhibiting important function. Now, if that's true, inhibiting the importans should reduce the levels of their cargo proteins in the nucleus. YAP, MYC, AR, transcription factors are key drivers of prostate cancer when they're in the nucleus. And so when you add 893, you can see that nuclear levels dramatically decline, presumably because KPNB1 and other important function is inhibited. Now, we made this compound because we wanted to mimic the effects of natural compounds like sphingosine and phytosphingosine. So I'm going to show you, yes, sphingosine actually does bind to these same targets and cause this similar conformational change to 893. The problem is if you put sphingosine into a cancer cell, it gets rapidly metabolized. So it's really hard to see its effects. But in these normal fibroblasts, you can see that both 893 and sphingosine dramatically inhibit the import of NF-kappa B into the nucleus in response to TNF-alpha. So this suggests that these sphingolipids inhibit nuclear import as well. So not only do they set up roadblocks here in the vesicular trafficking pathway, they also set up a roadblock into the nucleus. These parallel effects will cause really robust and profound changes in cell growth and metabolism. And that could make them very useful therapeutics. So if we activate PP2A and disrupt vesicular traffic, we can starve cells and make their mitochondria function better. I didn't really talk about this, but PP2A also turns off pro-growth signaling, and so that can be beneficial to decrease nutrient demand and growth. KPNB1 controls the nuclear import of many important transcription factors that promote growth, inflammation, fibrosis, so it's got a critical role in the cell. And these guys, important 7, transportin 1, and important 5, also important things to the nucleus that are important. But what's interesting is they have functionally overlapping roles with KPNB1. So c-Jun, which is another transcription factor, and ribosomal proteins can use any of these 4 importans to get into the nucleus. So if you want to stop their nuclear import, you need to inhibit all 4 of these importans. So I think that provides a potential explanation for why sphingolipids would inhibit these 4 importans. Sometimes they work on the same proteins. Okay, so I focused on the implications today for cancer and obesity, but excessive fragmentation of mitochondria causes lots of problems, including neurodegenerative disease, drug addiction. There have been studies in mice where you make mitochondria more tubular. You can fix neurodegenerative disease and drug addiction. That was a pretty impressive study in cocaine-addicted mice. In addition, it could be useful to increase oligonucleotide therapeutic delivery. So nucleic acid therapeutics are the future, right? Most of us sitting in this room have been injected with an RNA vaccine. I think the future of medicine is bright. The Nobel Prize this year went to a pair that made this kind of science possible. However, there is a problem with nucleotide-based therapeutics. Nucleotides are also negatively charged. Many of them are 5 to 15 kilodaltons. They cannot get across the city wall. They cannot cross the plasma membrane. So they have to enter by endocytosis. Once they get into the cell, we talked about how those endosomes can be recycled right back out. In our studies, we found that 80% of the oligonucleotide is immediately recycled back out of the cell. If they do manage to stay in the cell, they're still locked in these endosomes. They need to get to their targets in the cytosol and nucleus. And if they get pushed onto the lysosome, escape is even less likely because the lysosomal membrane is designed to block breaches. So in the end, only about 1 to 3% of the therapeutic that gets put into your body reaches its target. That's very inefficient delivery. So I think everybody in the field would agree this is the major barrier to using oligonucleotide therapeutics. Other people in the field have shown that this recycling endosome and multivesicular body are the sites where natural release of oligonucleotides is most efficient. And I showed you that 893 puts up roadblocks that traps things in those two compartments. So because there's lots of fission and fusion there, vesicles are pinching off, there's little holes that the oligonucleotides can sneak out through. So we thought maybe 893 would increase the delivery of oligonucleotides to the cytosol. So we tested this. We're using a fluorescently tagged antisense oligonucleotide. And you can see that it's brighter in the 893 treated cells, but it's kind of hard to see the cytosol because the endosomal signal is so bright. So we trapped the endosomal signal in an ROI, a region of interest, deleted it from the image, and then we optimized it for viewing, and we quantified. And you can see here, there's about almost a 10-fold increase in the amount of oligonucleotide that escapes into the cytosol. And that translates to a truly exciting increase in activity. So this is an antisense oligonucleotide that decreases the cellular level of the long non-coding RNA mallet1. And adding 893 shifts the dose response curve 2 logs to the left. That means it increases the activity of the antisense 100-fold. This was not in a specific cell type. Every cell type we tested responded to some extent. These were all done with a particular chemistry and target of ASO, so we could do apples to apples. But we did test it with other chemistries, siRNAs, other targets. It potentiates lots of different kinds of antisense therapies because it helps escape. It's not really specific to what you're putting in. It's about cell biology. This actually works in mice as well. The example I'm showing you here is an antisense oligonucleotide targeting a sodium transporter called ENaC. And this is a target to treat cystic fibrosis, so it needs to be active in the lung. We gave our mice 893 and treated them with a high dose of this ENaC oligonucleotide, and you can see it increased the activity 3-fold. When we used a low dose that's not even active on its own, 893 allowed it to be active. So that was an exciting finding. The brain is really rich with targets for oligonucleotide therapeutics. Everybody is really excited about the brain, but they don't cross the blood-brain barrier. Now, 893, I showed you, changes mitochondrial shape in the brain. We've done mass spec and confirmed that the compound gets into the brain. It crosses the blood-brain barrier. And here we're using a special kind of ASO. It's a cholesterol conjugated duplexed oligonucleotide. These get into the brain a little bit, but we were able to make them work a lot better in several areas of the brain. So in the brainstem, the spinal cord, the hippocampus, there's a trend in the cerebellum. Cortex was kind of disappointing, but I think that's more about the oligonucleotide and not about the compound. Further work needs to be done there. But it's an exciting possibility that by manipulating cellular traffic, we could improve oligonucleotide activity and make some new therapeutic treatments possible. The last thing is many viruses also, their life cycle depends on trafficking through the cell. And we've actually seen that 893 reduces coronavirus levels in the lungs of infected mice by 99%. So it's pretty impressive. Maybe viral diseases are on the horizon too. The point is, if we can make this into a molecule, convert this lead to a drug, there are many possible applications for compounds that work like this. Okay, I'm gonna end here by answering the most frequently asked question to the best of my ability. How is this not toxic? How is something that does this not killing everything? It's kind of surprising. And I've thought about this a lot. These are the best answers I can give. So the first one was, this is not my idea, right? We did not set out to make a compound that hits these targets. We made a compound that mimics the phenotypes of sphingosine and then figured out what it does. It's evolution, nature, that picked this constellation of targets, not our idea. Second thing is, it does all these things at once, and you think, oh, doing all those things at once, it's going to make it more toxic. But actually, if you think about it, all of those things at once are creating a program for quiescence and slowed metabolism and slowed growth. So maybe doing all those things together actually keeps supply and demand coupled and is less toxic. And finally, the natural sphingolipids are turned over, as I showed you, and 893, it doesn't last forever. It gets excreted and its effects go away. Its half-life is 10 hours. So the normal cells don't have winter all the time. They have winter and then spring, summer, fall to recover. When we're treating the mice with obesity, we're treating 3 days a week. I think if this compound was delivered continuously through an IV drip, it would be toxic. Okay, summary of everything. Sphingolipids disrupt intracellular trafficking. They can restrict access to nutrients and make your mitochondria work better. This means that they can fix cancer, fix obesity, at least in mice. The mechanism of action is kind of unique and I think kind of cool. They activate protein phosphatase 2A and inhibit importance that chaperone things into the nucleus. And these parallel actions have really robust ability to reprogram normal cells and kill cancer cells. There's also a potential we can use compounds like this to make nucleotide therapeutics work better, which is really exciting to me. And I'm hoping that we'll be able to avoid toxicity that often limits drug development because we're copying a natural compound that was optimized by evolution. Okay, so now this is probably my favorite slide since I'm talking at UCI. I'd like to thank everybody in my lab who's worked on this project. All the people in yellow have moved on to other pastures. I want to point out Rebecca, Cameron, Natalie, Warren, and Andrea. I think all of them are here. They've been working on the target identification, validating the importance as targets. Rebecca Lim, Brandon Chu, and Alexa are the macropinocytosis team, and I didn't talk about that today because it didn't really fit into this story, but it is a very cool story. And then Brandon, Alexa, Brendan, and Ariel are all working, and Raul, are all working on the antisense oligonucleotide angle. And I have lots of collaborators here at UCI that made this possible, and since some of them are here, I'll point them out. So Selma helped us use the metabolic cages. Angela let us borrow brie to gavage the mice during COVID. Karina and Guidre helped us stain brains for mitochondria, which was much harder than we thought it would be. Adila has trained just about everybody in my lab to do confocal. Bruce and Raquel let us use their echo MRI to measure fat and lean mass. Steve has been my long-term chemistry collaborator for about a decade. Peter's here, and Da Wei. They made a cell line, super cool cell line that's lacking the PP2A scaffold when you add a chemical called auxin. That was really helpful to validate our results. Chul Soon and Sun Hee have been helping us measure sphingolipid levels in cells. David Mobley and his team are working with us to model how these small molecules interact with the targets. Celia introduced me to the thermal shift assay and let us use her instrument for a long time. Carl Kottman lent us the cages with the running wheels, which I think made a really nice point in the study. Katrina has taught so many people in my lab how to use mice properly and be quiet in the mouse room. Wen-Chi is the best neighbor ever. I can't say enough nice things about Wen-Chi. I've known May for 25 years. We both work on PP2A and metabolism, and she's a great colleague. David Fruman has helped to support these studies. He's also been the conflict of interest person on a lot of my trainees' proposals, and that's been super helpful, not a super fun job. And if I forgot anybody, I'm sorry. The collegial environment at UCI is what made all this research possible. So thanks for coming. Happy to answer any questions.
That was great, and obviously the translational implications are profound. So I guess I'll start off and ask the first question. When you see the reversal effects after you administer 893, particularly on the mitochondria, is there a minimum conversion that has to happen for you to see a biological effect?
So you're asking a very good question. However, that obesity study was done with one $30,000 ICTS seed grant. The whole study. So I never had a grant. I was stealing from other places, unrestricted funds, right? So that study wasn't done as thoroughly as I would have if I had unlimited funds. So we don't know. We knew that the maximum concentration of 893 was in the blood 4 hours after we gave it orally. And so we had to fatten the mice for 6 months, which is really expensive. And then to look at their mitochondria, we had to kill them. So that study is really hard to do a lot of different ways. Plus, there's a circadian component because mice are nocturnal. So we had to give the compound at 3 in the afternoon because the lights go on in the mouse facility, off in the mouse facility at 6:30. So we wanted it to have peak levels right when the mice start eating. And then sometimes they had to sack the mice at 11 p.m. and go do all the microscopy. So, I don't have a good answer except it was hard and we didn't have any money.
Right. Okay. All right, and so let's open it up to the audience, okay, there in the back, in the middle. Right.
That is a great question. So,
can you repeat the question?
I was just about to, thank
Okay,
you.
thank you.
So does Ozempic, which is a GLP-1 receptor agonist, have the same mechanism as this? So they have the same mechanism in that they both suppress appetite, but they do it in different ways. So GLP-1 is not the same as leptin signaling. Insulin also regulates appetite. So we think that we'll work synergistically or better when we combine with Ozempic. So I actually have my company has a grant from the NIH to study whether 893 works better in combination with semaglutide, which is the active thing in Ozempic. So that study is underway. We also think that the GLP-1 agonists won't do as good a job stopping heart disease and liver disease as we will, because we have a mechanism that will directly stop inflammation, directly stop fibrosis. It won't just be the weight loss, we're directly targeting those bad things. So we're hoping we'll be better than Ozempic, because if we're just as good, we're not going to raise the money we need to get to the clinic.
Peter.
Aimee, that was a great talk. Thank you. I have 3 questions. The fact that
Only Peter.
the fact that drug isn't already in use and you're a multimillionaire and the school is not named after you implies to me that there's something wrong with it.
Reasonable question. Can I answer this question before you ask the next one?
Sure.
Okay, so sphingolipids, like this one, are outside of what's called standard drug-like space. There's this analysis, if there's a chemist here, they'll probably mock me because I'm not, but you can measure the Tanimoto coefficient. And these guys are way off the mean, okay? So we can't really use artificial intelligence to optimize it because these are so weird, okay? At the same time, FTY720 is very similar to 893, and it's a drug in the clinic. So, I think if we want to do new things, we have to go to new places. So no, it's not traditionally drug-like, but our preliminary data suggests we can get there.
But I've got a second part to my first
Okay.
question. What does it do long-term to mice?
Well, we've only given it for 3 months, and they look totally fine. We actually in the mouse room, we grabbed somebody from another lab and said, come look at these mice and see if you can tell me which cage is treated. And they couldn't. They compared the normal mice and the treated mice. So, the problem is actually related to its solubility. And I think we know where we can add some polar atoms to increase the solubility. So that's kind of where we're going. I mean, you'd think it would kill them, but it doesn't. It really doesn't. We've given it to dogs. It's not like we give it to rats and they're fine too until you give too much, and then it precipitates in their stomach and causes an ulcer, and that's not good.
So the second question is about basic biology. The fact that it targets the PPR128 subunit and then the importans, do you think you could modify the molecule so that it targets either the PP scaffold or the importans in order that you could distinguish the effects of those, but also to have a drug to selectively inhibit those different molecules?
So I would love to be able to do that. And actually, we have a library of these compounds, like 150 in the freezer. And my daughter, Natalie, is actually doing thermal shift assays on them for her high school project. So hopefully we'll have an answer for you that will find some molecules. It already looks like there are some that really shift the conformation of KPMB1, but they're okay on 2A. So I think we're going to be able to separate those activities. That would be very cool.
And my final question, I promise, is the sphingolipids, as you said, are natural molecules. If chemists look at 893, do they see any natural molecules that might look like that?
Well, sphingosine. So like sphingosine 1-phosphate. People have been trying to drug sphingosine kinase for years. Like multiple pharmas had big programs for drugging sphingosine kinase. We got compounds from them. Everyone's looking at this the wrong way. They're all focusing on S1P. It's not about S1P, it's about sphingosine. But the problem is sphingosine can go to S1P, sphingosine can go to ceramide. So if you don't lock it here, it gets converted, metabolized to other things, and you can't see the effects. So we have a compound that's locked in that sphingosine mode. And that's why now we can see what's going on. So actually, a paragraph in the paper is like, why hasn't anybody seen this before? It's because they weren't thinking about the problem the right way.
But is there a natural molecule that looks like
Sphingosine. And phytosphingosine. Those are compounds that are present in your body and mine and yeast.
Okay, Karina?
That was a great talk. Really
Thanks.
Very clear. I have a couple of questions about the obesity stuff. One is how plastic are the mitochondrial forms? In other words, if how often do they shift between these meatball and spaghetti shapes?
So that's another question. If I'd had more money, I would have loved to answer. So there are a few papers where they look at the circadian rhythm of mitochondrial shape. And I think that's a really important component. Like as we're planning this study to test against semaglutide, we're instructing the CRO, they have to be treated at 3 p.m. if your lights go off at 6, stuff like that. So I think, we have to think about how the biology is changing over time as well as our drug metabolism. So I don't know. In response to a meal that's got a lot of palmitate in it, so a bacon cheeseburger, your mitochondria are going to fizz. And that's okay. Right? Like in the old days when we were out running across polar bears and ate the liver and there was lots of fat and that was great. You fragment your mitochondria to deal with all that fat briefly and then they go back to normal. But now we're doing that all the time and our biology is not meant for the kind of continuously rich diets that we have. So I think the etiology of obesity is complex. Genetics, environment, exercise, biology.
Thanks. And the other part of it is for the mice fed on the high-fat diet who got the drug, did they lose weight because they didn't eat, or did they lose weight because they ate but their metabolism was higher?
So that is an important question. They lost weight probably because they ate less. So because we restored leptin signaling, they didn't eat as much food. But we also had a pair-fed group. So you can run another group behind the ones that are getting the drug, and you feed them whatever your treated mice ate the day before. Pair feeding does not correct leptin. We didn't get a chance to see if parafeeding corrects the mitochondria, but if it doesn't correct leptin, it's probably not fixing the mitochondria. So this isn't just about eating less. That's a great question.
David?
That was a great talk, and I love the city metaphor. As an immunologist, I have to ask two questions. The TNF result was really interesting. I haven't seen it before. Have you measured the effect on inflammation?
Now, David, that figure is in the paper that you are on, so you have seen it before, you just don't remember it. But your question is what?
You had to say that.
You just set me up for failure asking me an immunology question. Okay, go ahead.
Yeah, so does it have effects on inflammation? And on the flip side, have you actively immunized mice, not with the nucleic acid vaccine, but a typical vaccine to look at T and B cell responses that usually require things like MYC to enter the nucleus?
Yes, it's very complex. What it's going to do to the immune system is a big issue with the cancer part of it, right? So there's some evidence actually that if you suppress MYC, you change PD-L1 expression so that you sensitize to ICB. So there are like ways it might make sense, but yes, it's an issue. For obesity, you want to stop inflammation. Inflammation is a driver. For COVID, for the coronavirus stuff, we didn't have money for that study either, so we didn't get to do the whole workup. But I bet it will also help COVID not only by stopping viral replication, but by stopping the immune response to the virus, which is what really makes you pretty sick. So I think we'll have to be careful about that. If you want to work on the immune system with me, I would love to figure out what this thing does to the immune system.
In the front?
Mm-hmm. I'll repeat it after you say it.
Okay, well, thank you for the talk. This is my first talk, and it was truly fascinating. My question would be, for people dealing with obesity and trying to lose weight, to what extent could this molecule be part of that weight loss?
So the question was, for people who are trying to lose weight, how would you see integrating this molecule into a weight loss program? And that is a great question that I've thought about a lot recently. So there's this I-Core program at Applied Innovation where you go out and you interview your customers, right? And if you do all this, they give you $5,000 for your company. So we've been interviewing physicians and patients and dietitians. And one of the things that I've really taken away from that is we need to integrate this with a mentored program that helps people improve their diet and their exercise regimens. Because I don't want people to take this drug for the rest of their lives. I want to help them correct their leptin resistance. Then they have a healthy lifestyle that they can stay at an ideal acceptable weight for the rest of their lives without taking drugs. And talking to people in industry, that's not the way they're thinking. Industry is not really thinking about diet and exercise. They're thinking about, you're going to take Ozempic for the rest of your life, right? And people who go off Ozempic, sometimes they gain more weight. So talking to people who are taking that drug, it makes them nauseous and sick, and there are a lot of people who don't like it. So I don't know whether our compound will do the same thing. By controlling appetite, it may control it in a different way. We'll see what happens. But I see us marrying lifestyle changes with medical therapy with the goal of weaning people off the medicine.
So, Aimee, let me ask the last question. And, probably one of the ugliest words in the English language is aging, right? And do you see this having any effect on that process?
There are so many experiments I'd like to do. So when Tamu was here and worked on yeast, I wanted to do an aging study. I had a postdoc who used to work on C. elegans. I wanted to do an aging study in C. elegans. Yes, I think this controls aging. There are lots of links between sphingolipids and aging. Yeah, so it's just a bandwidth issue. I think it would be very interesting to look into that if there's anybody in the audience who wants to collaborate. We have plenty of compound to share for good experiments.
Okay, let's thank Professor Edinger again for a truly terrific talk.